The vaccine in that syringe has no virus in it. No live virus, no dead virus, not even a piece of one. It's something completely different, a message. And that message is why it works. Let's take it completely apart. The message is a strip of genetic code called mRNA. It carries the recipe for exactly one harmless piece of the virus. For COVID, the spike protein on the outside. Your own cells will read it and build that piece themselves. The vaccine never gives you the threat. It only gives you the description. But here's the catch. Naked mRNA is incredibly fragile. Enzymes called RNases are everywhere in the body, and they'll shred it in seconds. And even if it survived, mRNA is a large, negatively charged molecule. The cell's outer wall is made of fat. There's no door in, so raw mRNA injected alone is a paper note thrown into a river, torn apart before it's ever read. The solution is the real engineering story. Wrap the mRNA in a tiny fat bubble about 100 nanometers across, roughly 1,000th the width of a human hair. That bubble is called a lipid nanoparticle. It's built from 4 different lipids working together. The key one is an ionizable lipid, neutral in the bloodstream, so it glides through harmlessly, but the trap it sets is coming. When a cell meets the lipid nanoparticle, it doesn't open a door. Instead, it wraps its own membrane all the way around the particle and pulls it inside, like swallowing an envelope whole. The whole package ends up sealed in a bubble inside the cell called an endosome. The lunchbox is locked. Inside the endosome, the environment turns acidic. That acid is the trap springing. It flips the ionizable lipid electrically positive. Now it grabs the endosome's own membrane and tears it open, and the mRNA spills into the cytoplasm, the cell's main workspace. Here's the brutal truth. Only a small fraction of the mRNA escapes. Most is destroyed in the endosome. Improving that escape rate is the single biggest research target in the entire field. In the cytoplasm, the cell's own ribosomes find the mRNA and read it exactly the way they read any of the cell's own messages. They assemble amino acids in order and build the spike protein. Here is the key detail. The mRNA never goes into the nucleus. DNA stays locked inside the nucleus, and there's no root in. There's one more secret, and it won a Nobel Prize. When scientists first made mRNA in the lab, it set off the body's virus alarms, an immune response that both inflamed and shut down protein production, so the whole thing barely worked. Katalin, Karikó, and Weissman fixed this by swapping one RNA letter, uridine, for a modified version called N1-methyl pseudouridine. That single change makes the message read as self, quiets the alarms, and dramatically boosts how much spike protein the ribosome makes. That discovery won the 2023 Nobel Prize in Physiology or Medicine. The spike protein the cell makes gets chopped into fragments and displayed on its surface, like posting a wanted poster. Immune cells notice it. B cells learn to make antibodies that lock onto that exact spike shape. T cells learn to hunt cells showing it. Then the mRNA breaks down within days, and the spike protein clears. It can't change your DNA. There is no enzyme here to convert RNA back to DNA. And even if there were, the nucleus has no transporter that lets mRNA in. The message self-erases. The training lasts. The COVID vaccine was just the first product off a platform. Because you only change the message, not the factory, you can in principle reprogram it for almost any target in weeks. 2 programs are now in phase 3 trials for personalized cancer vaccines. Moderna and Merck's mRNA 4157 for melanoma, and BioNTech's autogene cevumeran for several cancers. These sequence a patient's own tumor, find its unique mutations, and encode up to 34 of them into a custom message. None are approved yet, but the factory now exists. If you want to keep following the frontier, subscribe. And next, how your phone is talking directly to a satellite. Katalin Karikó spent 15 years trying to prove this was possible before anyone gave her a grant to do it. She joined the University of Pennsylvania in 1989 with one goal: make mRNA medicine. Every grant proposal was rejected. Her university demoted her. She lost her faculty position and worked at a reduced salary in a basement lab, still convinced the idea was real. Nobody wanted the problem she was solving. In 2023, the Nobel Prize in Physiology or Medicine went to Karikó and Weissman. It was 34 years from the idea to the medal. The problem Karikó was fighting is called innate immune activation. When scientists first injected synthetic single-stranded RNA into cells, the cell's immune sensors, proteins called toll-like receptors, recognized it immediately as foreign. Foreign RNA means virus. The cell flooded itself with inflammatory signals and shut down protein production entirely. The very mechanism the mRNA was supposed to trigger, ribosomes reading a message, was switched off by the alarm it caused. The molecule worked perfectly in a test tube and destroyed itself the moment it entered a living cell. Making mRNA starts with a DNA template, a short synthetic strand carrying only the sequence you want expressed. An enzyme called T7-RNA polymerase reads that template and assembles a complementary mRNA strand, one nucleotide at a time, in a process called in vitro transcription. The reaction runs at 37 degrees Celsius in a biochemical bath. A few hours later, you filter and purify the mRNA away from the template and the leftover enzyme. What you have is billions of identical copies of the same molecular instruction in a test tube ready to load into lipid nanoparticles. Karikó and Weissman's discovery came from a systematic experiment. They made dozens of versions of the same mRNA molecule, each with a different chemical modification on one nucleotide. Then they exposed each version to human immune cells and measured the inflammatory response, how loud the alarm was. Version after version still triggered the alarms, until they reached N1-methyl pseudouridine, a modified uridine that occurs naturally in human transfer RNA. The cell reads it as self. The inflammatory response collapsed to nearly zero. It was a search through 96 chemical variants to find the one that made the immune system stand down. Here is the number nobody talks about in the mRNA vaccine story. Of every 100 mRNA copies sealed inside an endosome, somewhere between 1 and 2 escape into the cytoplasm before the endosome destroys the rest. 98 to 99% of the mRNA a cell swallows is degraded before it ever reaches a ribosome. The vaccine works because those 1 or 2 copies that escape are enough to produce the spike protein and trigger an immune response. But improving that escape rate from 2% to even 10% would multiply the effective dose 5-fold. It is the single largest research target in LNP engineering today. Once mRNA escapes into the cytoplasm, the clock starts. The modified mRNA in a COVID vaccine typically lasts between 6 and 12 hours in a typical muscle cell before RNases degrade it completely. The lipid nanoparticle protects it in transit. The pseudouridine modification extends its working life. But once the ribosomes have read it and the message is spent, the cell's own machinery recycles it like any expired transcript. The spike protein the cell makes lasts a few weeks longer. Immune cells keep finding and clearing it. But the mRNA instructions are long gone within a day. The mRNA sequence alone is not enough. 3 more engineering choices determine whether ribosomes can read it reliably. At the front, a 5' cap, a modified guanosine nucleotide, signals ribosomes to start here and protects the message from degradation. At the back, a poly-A tail, a long string of adenosine repeats, acts as a degradable buffer that gives the ribosome extra time. Flanking both ends, untranslated regions borrowed from stable proteins like globin tell the cell how many ribosomes to load simultaneously. The COVID vaccine optimized all three. Without them, the message is read once and destroyed. A personalized cancer vaccine begins with the patient's own tumor. Surgeons remove a sample. Genome sequencers read the DNA of both the tumor cells and the patient's healthy cells and compare them. Mutations present only in the tumor, called neoantigens, are identified. The ones most likely to be visible to T cells are ranked by computational models. Up to 34 are chosen and encoded together into a single mRNA molecule. That custom molecule is manufactured in roughly 6 weeks. The patient is injected and their immune cells learn to recognize those exact tumor signatures, a vaccine made for one person. In 2023, Moderna and Merck reported interim phase 3 data for their personalized cancer vaccine mRNA 4157, given alongside the immunotherapy drug pembrolizumab. In patients with resected high-risk melanoma, tumors surgically removed but at high risk of returning, the combination reduced recurrence or death by 44% compared to pembrolizumab alone. That is not a cure, but 44% fewer recurrences in patients whose cancer had already been removed is a number oncologists had not seen from that class of drugs before. No personalized cancer vaccine is approved yet. That phase 3 result is the first hard evidence the platform can move tumor outcomes. The next disease targets are ones where vaccines have failed for decades. A universal flu vaccine encoding dozens of influenza variants simultaneously, so a single jab covers every strain the virus might mutate into. An HIV vaccine, which has failed for 40 years because the virus mutates faster than any antibody can track. mRNA can encode dozens of variants at once. Treatments for rare genetic diseases where a missing single protein causes lifelong disability. Monthly mRNA doses that deliver a temporary replacement without touching the genome. The COVID vaccine proved the factory works and can be reprogrammed in weeks. The question is what we put in it next. If that made it click, hit subscribe. A fresh teardown drops every single day.